Dear students, we go on today with the second endocrine lecture. We continue with the hypophysis, with the thyroid axis. The diseases of the thyroid gland are among the most frequent diseases of the endocrine systems, and the history of thyroid diseases is well known back from thousands of years, and but today we have plenty of mobile phone applications also to manage these kinds of disease. But of course, one of the most important points in endocrine medicine is chemistry, which has brought up a lot of treatment methods, namely hormonal treatments, and the other important part in clinical endocrinology is surgery, where surgical technologies are able to cure different kinds of tumor diseases of endocrine glands. We move on now with the thyroid axis, and on this infographic we can again see the components of thyroid regulation, the hierarchical relationship of the different parts of the system, and there we see that the highermost level of regulation is the hypothalamus. From the hypothalamus, the TRH producing neurons will release their neurohormones in the median eminence, and through the portal circulation these factors will reach the anterior pituitary, where the thyrotroph cells will produce the TSH hormone, which then gets into the systemic circulation and then affects the thyroid gland itself. The thyroid gland, a peripheral endocrine gland, will produce its hormones, the T4 and T3, thyroxine and triiodothyronine, which then in the peripheral circulation will have their effects on target tissues. But on this infographic we can again see the negative feedback regulation, which is a key concept of endocrine regulation. In this case, T4 and T3 have feedback regulation on both the pituitary and also the hypophysis, the hypothalamus. On this chart we have also another kind of regulation shown, which is not related to the axis which I have explained now. Here on the right side we can see that the pars tuberalis of the anterior pituitary gland is involved in a TSH driven regulation of seasonal rhythms, seasonal rhythmic patterns of physiology. This is mostly prominent in animals, but today we also already know that this can have effect on the seasonal variation of mood and mood-related diseases, especially in winter time. The wintertime depression is related to this system. The anatomy of the thyroid gland, you have already got familiar with the topography of the thyroid gland. On the neck it is found beneath and in front of beneath the larynx and in front of the trachea. The two main lobes and the pyramidal lobe may be present in front of the larynx. And in a cross-sectional cut you can see the relation of the thyroid gland to the fascia systems of the neck, to the pretracheal fascia, and then a very close relation to the right and the left recurrent laryngeal nerves, which you have just behind the thyroid gland. Under certain not very rare conditions of the thyroid gland, when the gland itself gets hypertrophied, the thyroid gland can compress the neighboring tissues, including the esophagus, and under certain conditions even the trachea can be compressed and can cause severe breathing symptoms. And again, here I have a link for a surgery of the thyroid gland, where you can see in real time or in real life how the anatomical structures look like in a surgery, these anatomical structures. When we have a look at the histological structure of the thyroid gland, we see a very typical pattern of the parenchyma of the gland, which is then separated by connective tissue septums to smaller lobules. The parenchyma itself consists of glandular cells which are organized in cyst-like or sphere-like units, which we call follicles. These look a little bit similar to that what we have seen in the hypophysis, in the pars intermedia of the anterior pituitary. With the bigger magnification, we can see that the follicle itself is lined by a simple cuboidal epithelium, which we call the follicular cells. These are the endocrine cells of the thyroid gland. These are the cells responsible for hormone production. In the lumen of the follicle, we have a homogeneous eosinophilic substance, which is called, in histology, it is called colloid. The name refers to a substance, a fluid substance or a jelly-like substance, which is very rich in proteins, and that the same term is used also in the hypophysis for the pars intermedia. What we can see outside of the follicle, in the interstitium of the thyroid gland, that it is very rich in vessels, in blood vessels, and some connective tissue cells. And what we can also see here, that the follicles themselves are in a different size, but this size difference refers to the three-dimensional the different sizes do not refer to the three-dimensional size, but rather to the two-dimensional projection of the cut plane of the different follicles. So if we look at this structure, this is probably a tangential cut of another follicle, like the cut plane. If it would go through here, we don't have the lumen in the cut plane. We cannot really differentiate among the cells of the follicle with this staining. Most of the cells are follicular cells, but some of the cells which are closer to the basal membrane, which may be closer to the basal membrane, they may be different kinds of endocrine cells, but we can't really distinguish them from the follicular cells. With this staining, we would need to use immunohistology to differentiate those special types of so-called parafollicular cells, which are responsible for other kinds of hormonal activity, which I will talk about shortly. About the follicular cells, about their hormone production. In this chart, in this drawing, what we see here is a small drawing about this segment of the picture. That means in the image on the right side we have the apical part of the follicular cells, and in the left side of the image we have the colloid. So we don't have in the image the basal side or the capillaries. So these cells are the follicular cells, their apical side, and this is the lumen with the colloid. What we see here, that the follicular cells will secrete through exocytosis into the colloid, into the lumen of the follicle, a protein which is called thyroglobulin. This protein is very rich in tyrosine amino acids, and what we see here, that these amino acids will undergo an oxidative reaction or a chemical reaction with iodine, which will get into the lumen again through the follicular cells, which cells take up iodine ion from the blood and then secrete into the lumen, where a redox reaction will produce the atomic iodine, and that will be conjugated to the tyrosine amino acid residues. And so as the result, we will have the same protein, the thyroglobulin, of which the tyrosine amino acid residues will be iodinated. And then upon need, upon stimulus, the follicular cells will take up from the colloid bits of this substance of the colloid containing the iodinated proteins, thyroglobulins, through endocytosis. And then the cells themselves will break down these iodinated thyroglobulin to individual amino acids, and the iodinated amino acids will be secreted as the hormones of the thyroid gland. So we call these hormones of the thyroid gland T4, T3, depending on the number of the iodine atoms conjugated to the hormone, depending on 4 or 3 iodines. And then we got to the chart of the physiological regulation of the endocrine gland, of the thyroid gland. There again we see the hierarchy clearly shown, and but here the target tissues are highlighted, which also shows here an extra level of regulation with an enzyme in the peripheral tissues. This iodinase enzyme is able to transform the individual forms of the hormones of the thyroid gland, T4 to T3 and other derivatives, which have different kind of mechanisms or affinity to the target tissues, but all of them show negative feedback to the anterior pituitary. What is highlighted here that the TSH shows a major the TSH is the key regulatory factor in this process. And now we can move on to the next hormone, which is still related to the thyroid gland, and that is calcitonin. Calcitonin is a very different kind of hormone and produced in not the follicular cells but in the parafollicular cells of the thyroid gland, which we are unable to show with hematoxyninosine stained slides. This hormone is a protein hormone or a peptide hormone, calcitonin, which is related to the calcium homeostasis of the body, which means the balance of the calcium level in the blood is a very strictly regulated homeostatic parameter, which is regulated by several hormones, and one of them is calcitonin. And the antagonistic hormone to calcitonin is then the parathormone produced by another endocrine gland, which is located behind the thyroid gland. The parathyroid gland are two pairs of very small pea-shaped glands behind the thyroid gland. So that's a posterior aspect of the esophagus and the pharynx, and then we see the posterior surface of the main lobes of the thyroid gland, and these two altogether 4 small structures are the parathyroid glands. You can see here the close topographical relation to the recurrent laryngeal nerve. If we look at the histology picture of the parathyroid gland, you can see a cell-rich glandular organ where we may see very thin connective tissue septums, some adipose tissue also, and the main parenchyma consists of relatively darkly stained cells, but this dark staining does not come from the staining of the cytoplasm. With the larger magnification we can see that these cells are actually quite small endocrine cells with relatively thin cytoplasm, which means this is a relatively cell-rich tissue. You need to be careful not to confuse these endocrine cells with lymphocytes, and so not to confuse this organ with lymphatic tissues. In the same organ, in the same endocrine organ, in the parathyroid gland, we can see that the majority of cells are these small cells, which we call chief cells, and then there is another cell type which can be found in a group or also isolated, these bigger pale eosinophilic cells, which are called oxyphil cells. The chief cells are responsible for the production of parathormone, and the oxyphil cells are not known what their real function is. So this we don't know what are they for. And again, scheme of the regulation of calcium homeostasis, where we see that the stimulus for the parathyroid gland for production of parathormone is not related to the hypophysis at all, which means the calcium level of the blood is the direct stimulus, and that will increase the parathormone levels, which will affect the kidneys, the bones, and the small intestine to reabsorb more calcium to increase the circulating level of calcium ions. The next topic is the adrenal gland and its regulation. And on this chart, on this infographic, we can see again the typical hierarchy of the endocrine regulation of this axis, where we see that the hypothalamic regulation by CRH producing neurons will stimulate the function of the anterior pituitary, where the corticotroph cells will produce ACTH hormone, which will then reach the adrenal gland, and there it will regulate the function of the cells of the cortex of the adrenal gland. And the main hormone is shown here, which is the cortisol, which affect target tissues, various kinds of target tissues. On this infographic, not all of the targets are shown specifically, only the immune cells are shown, which reflects the medical importance of the hormone cortisol, that one of the most important medications today we can use against inflammatory diseases to calm the immune reaction, the immune system of the body, is cortisol. So there is a very important inhibitory effect of cortisol on the immune system. However, the main function of the cortisol is to affect metabolism of the body. That means carbohydrate or lipid metabolism, and all the affected tissues are target tissues of that hormone. And many other parts of the brain are also target points of cortisol. The anatomy of the adrenal gland, you have already seen the location of these glands in the previous semester. They are right above the kidneys, and their blood vessels show also an asymmetry because of the asymmetry in the great veins, or the big vein of the abdominal cavity, inferior vena cava, and the renal veins. Again, there is a link pointing on an operation of the adrenal gland. The histology structure of the adrenal gland is very typical. We can see here a parenchymal organ which can be clearly divided into cortex and medulla. There is a very clear border between the cortical part and the medullary part. When we have a look at the cortex itself can be also divided into three regions based on hematoxin staining, which is very useful to show difference in cellular density or differences in density of cell nuclei. And that is also the reason here why we see difference in the colors of different layers. The top layer is called zona glomerulosa, then the thickest middle layer is zona fasciculata, and then the deepest layer is the zona reticularis. These cells show different thickness of cytoplasm, and also the cytoplasm itself shows slightly different staining. We will discuss them one by one. But on this drawing we can also see, on this slide we can also see a drawing of the blood vessels of the cortex, which means that the arteries reaching the cortex will capillarize in the cortex, and some of them recapillarize again in the medulla. So here, this is the border of the cortex and medulla, and this capillarization is actually found in the medulla. That means hormones produced in the cortex will reach the medulla in a relatively high concentration, and this high concentration of cortical hormones in the medulla will have also an important effect on the medullary cells producing their own hormones. In the cortex, zona glomerulosa is the outermost layer, where we can see that the cytoplasm is very thin and very pale. Therefore, the cells look very dense in this area. The zona fasciculata, in contrast, has thicker cytoplasm. The cells here have much thicker cytoplasm, and the cytoplasm is kind of foamy. It contains many small bubbles which are unstained, which reflects the capacity of these cells to store lipids, to store fat, which is needed or which is used actually for hormone synthesis, because these cells here are producing steroid hormones, which means these are lipid hormones which are produced here. The zona reticularis is again a layer of cells where the cytoplasm is thinner, although the staining is a little bit darker or more intense in this case. This layer, zona reticularis, contains cells which are active in a certain period of the lifetime, not throughout the whole life. Are they very active? Their activity changes with age, and their function is most prominent during fetal development and in early life. But later on in adulthood, the function of these cells is a little bit more suppressed, or it's more in the background. Under pathological conditions, when the function of these cells increases, that will cause visible symptoms, because this layer produces sexual hormones in both genders, and the sexual hormones are androgenic hormones, like responsible for male phenotype. So in women, this layer is, though active, but it produces very limited amount of androgens needed for growth or some metabolic processes under physiological conditions. In the adrenal medulla, we see a very different pattern of organization of the cells compared to the cortex. The medulla is very rich in sinusoids, in blood vessels, which will drain the blood of the cortex as well, as I showed here in the drawing. And the cells form a network-like organization, parenchyma, without any special structures. The cells themselves are called chromaffin cells. The chromaffin name comes from a histochemical procedure which reflects the biochemical process of hormone synthesis in these cells. The medullary cells produce adrenaline, an amino acid derivative, which belongs to the molecular family of catecholamines. In this drawing, three of the hormone producing areas is highlighted. Two out of the cortex, the zona glomerulosa, the zona fasciculata, and the third one is taken from the medulla, the hormone producing cells of the medulla. So these are the hormone producing cells shown. Lipid hormones producing cells are relatively rich in smooth endoplasmic reticulum, and also the relative high level of mitochondria are needed for the biochemical reactions which are needed in the steroid hormone synthesis. In this drawing we can also see the key signals which stimulate the function of the endocrine cells. Zona glomerulosa cells are very special because they are not stimulated by hypophyseal hormones. Their functional regulation is by the blood level of other hormones not produced in the hypophysis, like angiotensin II or certain ion. The level of certain ions in the blood have direct effect on these cells. The output, the hormone produced by these cells, is called aldosterone. Therefore, this layer, the zona glomerulosa, we call the mineralocorticoid producing cells. The next one, in the zona fasciculata, cells, they are stimulated by ACTH, so these are the key cells in the hypothalamo-hypophyseal adrenal axis. These cells produce cortisol, which I have already mentioned, one of the most important hormones of the body. Without this hormone, people die after a certain period of time because their metabolism cannot adapt to any stressful conditions or any threats like immunological threats or whatever. So cortisol is a very important hormone for the normal physiology of the body. The adrenaline producing medullary cells have a very special regulation. They are not stimulated directly by hypophyseal hormones. They are mainly stimulated by sympathetic nerve fibers directly. So here we have a kind of neural regulation of endocrine cells in the periphery, which is kind of very special. And as I said before, hormones produced, especially cortisol produced in the cortex, will flow through the medulla as well, and that can have also an effect on the production of adrenaline. Now, some charts I'm showing here about the physiological regulations affected by the adrenal gland, especially by mineralocorticoids, so the zona glomerulosa cells. The zona glomerulosa cells produce aldosterone here, and in this system we see a key function of angiotensin II, which hormone will affect the production of aldosterone. And this angiotensin II is related to the blood pressure regulation. And how angiotensin II is produced is shown in this chart, which you have already learned in the topic of the physiology of kidney in the last semester. So this layer of the adrenal gland is not under the regulation of the hypophysis, but it is under the regulation of those systems responsible for the blood pressure. This is a summary of the regulation of the adrenal gland, where we see on the left side the regulation of the medulla, and on the right side the regulation of the cortex. ACTH is the key regulatory hormone from the hypophysis, which regulates mainly the production of glucocorticoids. It has effect also on the sexual hormone, the androgenic hormones of the zona reticularis as well, but their importance is not very prominent in adults. But here on this chart there is a mistake. Actually, ACTH has no receptors in zona glomerulosa, so the aldosterone production is not affected by the ACTH. And on this drawing we have a better picture of the regulation of the medulla, which we have seen here on this side. It is the same, which means hypothalamic centers for autonomic regulation are connected to the intermediolateral nucleus of the spinal cord, where the preganglionic neurons send their axons directly into the medulla of the adrenal gland. That is a very special space, a very special relation here, because preganglionic neurons do not synapse with a ganglion cell, a nerve cell, but they end directly on a glandular tissue, endocrine tissue. This is related to the development of the endocrine cells of the medulla, which are developing from the same stem cells as ganglion cells, sympathetic ganglion cells of other parts of the body. Later on you will have lectures about the autonomous nervous system, where you will learn in more details about the autonomic neural regulation of the body, including the regulation of the adrenal medulla. Okay. The next part of the lecture is a summary of the development of the endocrine organs. I have made slides for each endocrine organ with very short descriptions, which is needed for the topic development of the endocrine organs. So you can read through on your own. I don't need to say them or talk about this now. And at the end of the lecture material, you can also find some slides about the exam questions we ask during the exams when you get histo slides of endocrine organs. There is a relative high chance to get an endocrine organ as a histo slide during the final exam, as we have many of these organs and many slides of them. So have a look at these questions. These are the key questions we ask in exam. And at the end of the lecture you can find some information about research and history of endocrinology and the relation of our department to this kind of research. So you can have a look at that. Thank you very much.